Alabama STEM Explorers is made possible by the generous support of HudsonAlpha Institute for Biotechnology, Southern Research, solving the world's hardest problems, the Hawley Family Foundation, Alabama Works, Alabama STEM Council, Alabama Mathematics, Science, Technology, and Engineering Coalition, Alabama Math, Science, and Technology Initiative.
I was looking at these roses, and I'm wondering, how do plants grow and reproduce? I mean, I know that you can plant a seed and that can grow, but are there other ways that new plants can form? I think I know a place where we can find answers. Come on, let's find out. Hi, welcome to Alabama STEM Explorers. I'm Sophia, and I'm here with Neil at the HudsonAlpha Institute for Biotechnology in Huntsville, Alabama. So I was just asking him, how do new plants get created?
That's a great question, Sophia. And there are really two broad categories of how you get a completely new plant. One that you and our viewers are probably most familiar with is when we actually have a seed and we plant that seed and it forms a new plant. And that seed comes from the combination of a pollen grain and an egg in a plant. So that process is called sexual reproduction, and that involves the flowering parts of plants. The second process is called asexual reproduction, and that doesn't involve having two different parents contributing pollen and egg. And you see that in some of the other plants that I've got up here. For example, so this is an iris plant. I love iris flowers.
They're very pretty.
They're really beautiful. But you can see I've got two little plantlets right here next to it. These have formed from the side, and we actually could separate those from the plant, and they would have all the roots and the leaves and everything to make a completely new plant. I've also got some lily bulbs here, and the lily bulbs over time actually grow and actually form a separate bulb that you can separate out.
Wow, so it like created its own twin.
It did. It created its own twin. So that didn't involve any parts of a flower. That's an asexual reproduction process. We could do the same thing with this ivy plant if we took a pair of scissors and trimmed off a piece of this stem and put it in water, or maybe added a little bit of a hormone called rooting powder, and it would form its own set of roots, and we would have a completely new plant that is identical to the parent.
So here with the flowers, we have reproductive cells like pollen and egg cells, but here, do we have reproductive cells?
In this process, there are no reproductive cells involved.
So how does it grow from a non-reproductive cell?
There's a set of signals that takes place in the cells, let's say when we clip the stem, that actually tell those cells to become something different. Instead of being leaf cells, to become root cells. And that actually sits at the heart of another part of asexual reproduction that I want us to talk about, called somatic embryogenesis.
Somatic embryogenesis.
It's a mouthful, isn't it?
Yes, it is.
Yeah, it's a process that happens specifically in the lab. I tell you what, you slide this over to the side and I'll pull the whiteboard around. Great. So let's talk about words. Somatic embryogenesis. Embryogenesis is the formation of a new plant, the beginnings of forming a new embryo, a new plant. A somatic cell is a cell that's not a reproductive cell. It's not a pollen grain that has a sperm in it or an egg cell. It's a cell like a leaf cell or a root cell. So somatic embryogenesis is building an entirely new plant from a part of the plant that isn't normally involved in reproduction. I'm showing you this example with a cotton plant, but we could take a little bit of the leaf and we could put those leaf cells on a petri dish and we could add some nutrients, and those leaf cells would stop behaving like leaf cells. They would actually become like this mass of cells. It looks a little bit like a funky head of cauliflower, but what's actually happening is that the cells are going through a process called de-differentiation. They are losing their identity as leaf cells, and they are picking up the identity as the cells that could form a brand new embryo that you could then plant.
So I know that cells such as stem cells can differentiate and reprogram to become different types of cells, but what is dedifferentiation and how does that occur?
Let's talk about a couple words you use. You just said stem cells, and we're not talking about like the stem of a plant. We're talking about a category of cells. Stem cells have the ability to form multiple different types of cells. Embryo cells have the ability to form roots and leaves and flowers. They're stem cells. Leaf cells or root cells have lost the ability to differentiate, to form multiple cell types. So this dedifferentiation is a process that allows a cell that traditionally has behaved as one type of cell to now behave as the ability to form lots of different cells. So let's talk about how that actually happens at the genetic level. I'll push this back, pull those recipe books over.
The food looks yummy.
I like to cook. I think you like to bake. Do I remember that?
I do.
So this is two identical cookbooks. They have the same set of recipes. And we often talk about how DNA is like a recipe. It provides instructions for the cell for how to make things. So you can think of all the genetic information in a cell as being its genome, like this recipe book. Now look at these two recipe books and tell me what is different about them.
Well, this one doesn't have anything in it, but this one has binder clips and post-it notes, kind of flagging certain pages and, not letting you get to these recipes.
Perfect. That's exactly what happens in cells. So where I've flagged recipes like this one, which is for a bacon burger with chipotle mayonnaise.
Wow.
Yep. This is a recipe that I come to often, that I use often. And I've also clipped off pages that have recipes that I'm not interested in. Our cells do the same thing at their DNA level. Now, they don't use little flags and binder clips. They use a different set of chemical markers, but they silence genes that they don't need. For example, a root cell doesn't need any of the genes that are involved in a leaf because it's not going to make leaf proteins.
So this whole recipe book is the genome, and then certain recipes are instructions, and they can either flag them or kind of set them to the side.
That's right. And the process of differentiation, making a leaf cell, puts all these flags and these specific binders on that shuts off parts of the genome. An embryonic cell looks more like this. It doesn't have any restrictions on it. It could turn every gene on it needed. So the process of dedifferentiation in a cell is taking all of these marks off and giving us a cell that looks more like this that can then form brand new plants. So I work with a group of scientists that are actually studying this dedifferentiation process in somatic embryogenesis, and they're looking at it in microgravity on the International Space Station.
Wow.
Yeah, because they want to know if different environments change the way this process happens. Would you like to meet them?
I would love to meet them. This
All right.
sounds out of this world.
I'll go get them. Sophia, let me introduce you to my friends and colleagues. Chris from Clemson, Jane and Jeremy, both from here at HudsonAlpha. And it's a great example of team science, the idea that multiple researchers work together on big projects. And they've brought some great props to talk about the cotton research.
This experiment, could you tell me a little bit about it, and how is it conducted in space?
So we're trying to understand what genes and how they're regulated in cotton plants in microgravity in space on the space station, as well as on gravity here on Earth. And we can compare these two experiments. So here's an example of what actually went up to space here. So this is basically stem segments that were cut from these seedlings that were germinated here on Earth. Here's an example of about 45 days where you can start to see dedifferentiated cells that are dividing, and eventually these dedifferentiated cells will reprogram and form an embryo. And that embryo will form a whole plant or develop into a whole plant.
So what's different in space than on Earth? Why can embryos form in space while they can't here?
Well, our hypothesis is that the program to go through the process of somatic embryogenesis is encoded in the DNA in most, if not all, plants, but it's heavily silenced here on Earth. So
just like the binder clips.
That's right, the binder clips. So space offers us a unique environment where with microgravity, it's been shown that these binder clips can be removed and reorganized in space. So we're testing to see what happens in microgravity when we conduct this program up there.
That's amazing. So after the experiment comes back from space, what happens?
We're so excited to sequence DNA and RNA that's been in space. So we receive the plants, we'll extract the DNA and the RNA, and then we'll then produce sequencing readout, which will give us an idea of which genes were expressed and where the DNA clips or binder clips were when the tissue was collected.
So how do you guys see where the post-it notes and the binder clips have been?
This is a specialized form of DNA sequencing where the DNA is modified and we read out this particular tag, which will tell us where those tags were. And all this sequencing is done very high throughput, so we don't do one sequence at a time. We do sequencing in these microtiter plates where you can sequence many samples at the same time. And then when we sequence, we do it in very high throughput sequencing instruments using these flow cells where all the sequencing is done within this one unit.
So all the science takes place within that
It does.
cell right there.
It does, and
that flow
produces
cell.
a huge amount of data all at the same time.
And you're looking at chemical marks. You know, we've talked about binder clips and post-it notes, but really you're looking at chemicals that are added in specific places on that DNA sequence. And so once you get all that information, Jane, then it goes to Jeremy and Jeremy's team to figure out how to interpret that. And how does that happen?
Yeah, well, my team works on computers. And so Jane mentioned that we produce a lot of data. So for example, this kind of flow cell here produces about 3 trillion DNA sequences every about 2 days.
3 trillion?
3 trillion, yeah, it's a big number. It's got a lot of zeros after it. I get confused myself how many zeros are there. And somehow we have to sort through all that data that comes back off the instrument and figure out what each of those individual sequences mean. And so in this context, it means using computers to go through and identify the tags, for example, and these DNA modifications, and be able to stack up a whole bunch of different sequencing reads, individual little collections of A's, T's, and G's, to figure out which ones of those base pairs are actually changed, where those binder clips are. And then for the expression data, which is the RNA data, that tells us when genes are turned on and when genes are turned off. And as we look across the time course of embryogenesis and plant regeneration, we then measure the number of times those genes are turned on or turned off using computers to match those genes to something called a reference genome.
So after the experiment gets back, what you do is you figure out what the words in the recipe book are, and then you figure out what's changed.
Right. I figure out what's changed in the experiment. So you do it. Everything is about comparing between 2 different conditions. And so in this case, we're comparing between microgravity and the same control plants that are grown on the Earth. And by comparing the 2 of those, we can then extract information about how the biology works and how those genes are turned on or off and how that affects whether we're able to reproduce these full plants back out the other side for these cotton plants.
So this is a really cool process, but why is it important?
So once we understand the genes that are involved in somatic embryogenesis, we can use biotechnology approaches to trigger those genes, and basically allows us to accelerate the breeding process to release new crops in a much faster way with more impact to agriculture. It also gives us opportunities and new ways to store species in the form of cells. For example, for deep space exploration missions, we could send up a diversity of plant species for astronauts to use to colonize other planets and things like that.
And it also allows us to rapidly improve plants for sustainability purposes. For example, improving water use efficiency or reducing the amount of fertilizers we use to grow plants out in the field here. And that makes a big difference for us moving forward as a species to be able to continue to improve our sustainability of our agriculture and its impact on the Earth.
That's a lot of impact for a really tiny set of cells. Sophia, we have one last set of people that I'd like you to meet, and that's the astronauts on the International Space Station that are actually doing the work. So we're going to head into that space so that we can have a conversation with the folks in space.
So how do you carry out the cotton experiment, and what specific things do you do?
Well, our job up here on the space station is just to take all of the plans, all of the work and the preparation of the scientists on the ground and the operators, and just execute the plan as they've designed it. So what we do is we put the plants in an environment that they can thrive in. We monitor them, we take pictures of them, deliver those pictures back to the ground so scientists can figure out how well they're growing, and then we preserve them and get them ready to come back down to Earth. Because we know there's a lot of work that has to be done on the plants themselves, looking at the genetics, looking at the growth rates. This is all very important for figuring out how plants grow in space.
Did you practice the experiment on Earth?
That's a great question. We honestly, the last time I had training in the United States was about 15 months ago. So, and we have so many experiments we get trained on. I can't say with certainty that I did or did not get trained on this particular experiment. I can tell you that the emotion that I had while working on the experiment was I felt very comfortable doing it. So whatever happened, I felt well prepared.
What have you guys enjoyed most about performing the experiment?
Well, being able to do such great research up here in space is a real privilege. We have science technical backgrounds ourselves, and so of course we are very curious about how the experiments are going to turn out. So to be there actually in the place in zero G where the plants are growing and to be able to see them with their own eyes is a real privilege and actually very exciting. And then being able to talk to the scientists, share emails through mission control and information about how they're growing is very exciting as well. So, and we enjoy having the plants up here. It enhances our life up here, both visually and just in knowing that we are taking some small steps to being able to grow plants in space so that many humans can be living off of the plants and enjoying them as well in the future.
What do you like most and least about living in space?
The thing that I like most about living in space is the sense of purpose I have. But I tell you, the thing that follows right after that is the wonderful people that I get to work with. So I've been up here with more than 20 different people on this flight, and I have enjoyed getting to live and work with every single one of them. And as far as the thing I enjoy least about being in space is being and it's not Tom. It is definitely not Tom. He's on the thing I enjoy most category. I would say the thing that I enjoy least about being in space is the amount of time I've had to be away from my family because of this flight.
What made you want to go into space?
I've wanted to go into space for about as long as I can remember. When I was 8 years old, I saw Neil Armstrong walk on the moon, which gave me an enormous sense of wonder, sense of curiosity about what was going to happen in the future and what are other heavenly bodies, planetary bodies like, what are they like? And I have a bit of a sense of adventure and curiosity. I want very much to know what would it be like to be in certain places. I love backpacking and climbing and going into remote areas. And space for me was the absolute most exciting frontier I could possibly imagine. But it wasn't until I began to be interested in math and science and engineering and wanting to work for NASA that I really got involved and loved the work that people do and loved working with the people that do research that is required for going into space. So all those things came together and made me want to fly up here and experience it for myself.
How long do you guys have to train before you can go into space?
I would say in some ways we've been training our entire lives. So like Tom mentioned, before he became an astronaut, he did lots of exploring and kind of pushing his own boundaries. Certainly I did that a little bit as well. Now, but officially I became an astronaut candidate in 2009, and I didn't launch for my first flight until 2017. And all that time was training. So that was, for me, it was 8 years. For many astronauts, it's as little as 2 to 3 years before they launch to space for the first time.
What advice would you give to kids who want to become astronauts one day?
Well, I think Mark put it pretty well. You need to start training today and do those things that astronauts need to do, which you can start doing today. Astronauts have to study. We're always reading about not only the next day's work, but when we come to NASA, we have to learn about orbital mechanics and how spacecraft work, all sorts of things. And so one thing that we can say that we're pretty good at is we learned how to learn. And that's one of the most valuable things you can learn now in school. So start doing that right away. It's helpful to put yourself in a position where you have to make decisions. If not now, then as you continue on in your education, joining clubs, doing sports, that sort of thing, where you are putting yourself in a leadership position. And that's something we have to deal with quite a bit up here as well. And then finally, just taking care of yourself, enjoying hobbies, getting enjoying life in as wide a breadth of life as you possibly can, simply because you'll be living in space with just a few people in a module, most likely, and you want to be a good human representative and be able to have good conversations and have your crew members enjoy being with you as well. So I would say you might want to get a technical degree, but maybe not. Spaceflight is opening up for so many people that they probably need lawyers and architects and artists and writers, all kinds of specialties here in space. So really the space is the limit for you. That is, there is no limit. Just fall in love with something and do it as best as you can.
Those are all the questions we have. Thank you guys so much for your time and the work you do. We all really, really appreciate it. Thank you guys so much.
It was really fun talking to you as well. Thank you.
It was nice talking to you.
Sophia, what an incredible episode.
This has been so amazing.
How often do you get to talk to astronauts?
Not very often. It was awesome to see how a stem of a plant can be cut up and then de-differentiate, and then those same cells can re-differentiate again and form a completely new plant. And it's crazy to see how such small cells can have a huge impact on sustainability in agriculture or being able to grow plants in new and different environments.
We covered a lot of science in this episode, including the concept of epigenetics, which is the specific way that the DNA in our cells silences parts of genes or specifically marks and flags parts of genes. And that whole process of epigenetics is what takes us to de-differentiated and re-differentiated cells. It's pretty amazing to think about how these tiny cells make such a difference in the plants that we see and the traits that we want.
Gregor Mendel would be very impressed.
That he would.
I have always been interested in science and space. One of my earliest Christmas presents was what I called a telescope. Looking back, it was a toy, but to me, it was a telescope. My job with the James Webb Space Telescope was mostly in testing both the components and the final telescope for thermal expansion. Like I said, this telescope is 6.5 meter mirror. That's about 21 feet. And on orbit for 14 days at a time, they want tip to tip that mirror to stay stable to 38 nanometers. That's 38 billionths of a meter. That's a nanometer is about a millionth of a human hair. So 21 feet can move 38 millionths of a human hair in 2 weeks. So everything has to be super low thermal expansion. So every piece that was used to construct it had to be thermal expansion tested to make sure that it was what it behaved it was supposed to be, and then when it was all put together, we had to do testing to make sure that the whole structure behaved as it was supposed to. And we did that mostly with light, interferometers, autocollimators, essentially using light to make measurements with things. And that's my role is optical metrology, is the name of the science. The most amazing thing about a new instrument like this is what you'll learn that you didn't know that you were going to learn. Just for the record, it is 3 times the diameter of Hubble. Here's the interesting thing, it's doing the same science that Hubble's doing. We're hoping James Webb has the light gathering power and resolution to see the first light of the universe. And of course, that will tell us things about the origin of the universe that right now all we can do is speculate. So it could fundamentally change. It'll either reinforce what we believe or tell us we've got to go a different direction about how the universe began.
Thanks for watching Alabama STEM Explorers. If you missed anything or you want to watch something again, you can check out our website at alabamastemexplorers.org. Maybe you have a question we could answer here on the show, and you might grab a cool t-shirt. Feel free to send us a video question or an email on our website, alabamastemexplorers.org. Thanks again for watching. We'll be back next week.
Alabama STEM Explorers is made possible by the generous support of HudsonAlpha Institute for Biotechnology, translating the power of genomics into real-world results. Southern Research, solving the world's hardest problems. The Hawley Family Foundation, established to honor the legacy of Brigadier General Everett Hawley and his parents, Evelyn and Fred Hawley, champions of servant leadership. Alabama Works, a network of interconnected providers connecting business and industry needs to a highly skilled and trained workforce. Alabama STEM Council, dedicated to improving STEM education, career awareness, and workforce development across Alabama. Alabama Mathematics, Science, Technology, and Engineering Coalition for Education, advocating for exceptional STEM education in Alabama. Alabama Math, Science, and Technology Initiative, the Alabama Department of Education's initiative to improve math and science teaching statewide.